Friends, I'm Tim Gavin, and AK fellow number 586. I will be talking about Vasomyo Crosstalk in health and disease. This is the intracellular communication between cells of the vasculature and the muscles. We see an intimate relationship between muscle, here in this cartoon with mitochondria, and the vasculature, here with capillaries of the microcirculation. This close proximity is important for facilitating delivery of oxygen, insulin, other substrates, and the removal of waste products. And there is a close relationship between mitochondria in the muscle and the number of capillaries, here being expressed as maximal oxygen consumption, having a greater maximal oxygen consumption, having greater muscle capillarization. But also that regulation occurs as a function of the size of muscle fibers. Here we are looking at muscle fiber size, whether they're oxidative or glycolytic fibers, and more oxidative fibers have more capillaries, and bigger muscle fibers have more capillaries, suggesting that there is a regulation occurring between muscle and the vasculature, and we refer to this as Vasomyo Crosstalk. In this cartoon, we've depicted how new blood vessels, in this case capillaries, are formed. They're formed from existing capillaries, here in white, and they start out by breaching of the capillary, breakdown of the basement membrane. Then we see a proliferation of endothelial cells. All blood vessels have a lining of endothelial cells, and these endothelial cells must proliferate, they must migrate towards some central location, and then they will actually form tube structures and become capillaries. We know that there are growth factors and other factors that are involved in this process. We have done extensive studies investigating the regulation of and the importance of one of these, vascular endothelial growth factor, VEGF, in the response at rest, with aging, with other chronic diseases, and in response to exercise. VEGF is a muscle-produced cytokine. It's produced by many cells, including muscle. And when it's produced by muscle and released, these are known as myokines. What we see here in this graph, where we looked at the response of VEGF in young and older men in response to an exercise training program, we see that acute exercise increases VEGF both in young men and aged men, but that in aged men, the response is about half of what we see in the young men. And that impaired response is related to lower muscle capillarization, also coincident in aged men. More recently, we've been interested in what are known as exosomes, and now referred to as small extracellular vesicles that are produced by most cell types. This is a diagram of how these exosomes are produced, and I won't go into the details of it, but it's safe to say that these exosomes, or small EVs, are produced through a coordinated process that are then eventually released into the interstitial space of the cell. And then in muscle, they can be released by muscle and eventually enter into the circulation where they can have endocrine effects. We're more interested, because of our interest in the muscle vasculature, that interaction, we're more interested in the paracrine regulation of small EVs on endothelial cell and muscle function. I will also mention here that these small EVs have a wealth of information contained within them, packaged in very specific manners and tissue-specific manners with microRNAs, messenger RNAs, proteins and lipids. This is a EM picture of the skeletal muscle niche, and what I want to highlight here is that here we have mature muscle with the nuclei labeled MN. We have capillary endothelial cells here labeled EC, and we have muscle stem cells known as satellite cells. And I wanted you to pay attention to the close proximity between these three cell types. And it is that close proximity that we believe is important and facilitates intercellular communication between these different cell types. In the lower panel, what we have are extracellular vesicles that we've isolated in the lab. This is from Yahui in our lab. That shows their size. They're somewhere in the neighborhood of 75 to 100 nanometers in size, and it gives you an idea of what they look like. We also see that these isolated vesicles do resemble or are consistent with exosomes because they express exosome-related proteins. These are surface proteins known to be expressed on exosomes. We then asked, do skeletal muscle-derived small extracellular vesicles participate in the regulation of capillarization? And in particular, as I mentioned before, we need endothelial cells to proliferate, to migrate, and to form tube-like structures. And what we see is that when we take these muscle-derived small EVs, they promote endothelial cell growth, they promote endothelial cell migration, and they promote endothelial cell tube-like structures. So muscle small EVs are able to promote in endothelial cells many of the features that we see in vivo with angiogenesis. We also see that there is very specific packaging, in this case, of microRNAs. In the top panel here, we have microRNAs that we isolated from within the muscle cells. And in the bottom panel, we have microRNA that we've isolated from the muscle-derived extracellular vesicles. And what we see in the muscle itself is an expression pattern here of muscle-specific microRNAs, microRNA 1, 133A, 206, and then very small relative expression of the angiogenic microRNAs. In this case, 130A, I'll highlight. Compare that to the expression pattern we see in the muscle-derived EVs, and you see a much different expression pattern of the muscle microRNA and a relatively much greater expression of angiogenic microRNA, which tells us that there is very selective packaging that results or is part of the components of these EVs. And if you've wondered what I mean by microRNAs or what do microRNAs actually do, I've grabbed a cartoon here from Khan Academy where we have microRNAs. They're RNA sequences of about 22, 23 base pairs. And what they do is they bind a messenger RNA, and that can inhibit or prevent the translation of messenger RNA. One way is by binding to the messenger RNA, they target it for destruction. Another way is that by binding to the messenger RNA, they create a loop here, and the translation machinery is unable to actually effectively translate the protein that would result from this messenger RNA. So microRNA are unique in that they actually can target multiple pathways and multiple pieces of any given pathway at a single time. So microRNA are very attractive in studying because they can affect multiple pathways at one time, as opposed to select proteins or lipids, which are much more pathway specific. So we then started to ask the question, well, if we make muscle dysfunctional, does that impact EVs, and does that impact muscle-derived EVs' effects on endothelial cells? So this is work that we just got accepted here from Zach Hediger and Chris Cargill in the lab, in which we took muscle satellite cells known as myoblasts, and we exposed them to oxidative stress, in this case H2O2, and we looked at the production of EVs. And what we see is small EVs, here labeled exosomes, are actually increased after oxidative stress of the muscle cells. So we're getting more of them, and it's a result of an increase in production of EVs, which is what all these gene expression data is showing us. The next question we wanted to know was, okay, so there's more EVs. Are they good or are they bad? And what we saw is when we took these muscle-derived H2O treated EVs and exposed them on endothelial cells, we see a transfer of senescence-related activity, here measured by beta-galactosidase. So when we take EVs from H2O treated muscle, the signaling that is inside the EVs induces senescence in the endothelial cells. We see impaired proliferation in the endothelial cells, and using a wound healing assay, we see impaired angiogenesis as well. So oxidative stress leads to impairment through EVs of endothelial cell function. We then ask the question, well, we're talking about crosstalk. Is it just muscle to endothelial cells, or do endothelial cells also impact muscle function? So we took endothelial cells, we incubated them in high glucose media, hyperglycemia. We took the media off those endothelial cells and exposed them to muscle satellite cells. And when we did that, what we saw was the muscle satellite cells proliferated at lower rates than when we use normal glucose medium. So by making endothelial cells dysfunction through hyperglycemia and taking that media and exposing it to the muscle cells, we see impairment in muscle cell function. And the cyclin D here tells us that's because we're not getting enough replication occurring, and it's not because using caspase-3 as an index of cell death, we do not see that it's related to accelerated cell death. So then we ask the question, what about the actual formation of muscles? So muscle satellite cells, myoblasts, can fuse together and form muscle fiber-like structures known as myotubes. And when they do this, they become multinucleated like muscle cells are, and they can actually contract. And so when we take the media from dysfunctional endothelial cells and put this on the satellite cell population, it impairs their ability to fuse. It impairs their ability to become myotube-like structures and what we see is the myotubes have fewer nuclei per myotube. So we're not getting as much muscle formation when the endothelial cells are dysfunctional. So what we've shown is muscle dysfunction impacts endothelial cells and endothelial cell function, and endothelial cell dysfunction impacts muscle function. The last piece of data I want to show you is some work that we've just completed. It's a small pilot study that we took a look at the microRNA that we derived from EVs released from human muscle, from lean or individuals with obesity, and then we looked at where do these EV microRNA target? What pathways do they target? And what we saw is compared to lean, the EV microRNA from individuals with obesity target anti-growth and pro-inflammatory pathways. So that could explain some of the growth retardation we see in muscle in obesity, and also some of the pro-inflammation that we see in obesity. Remembering that EVs can have both paracrine and endocrine functions. And then as part of this study as well, we exercised a group of subjects and looked at pre to post 1 week of concurrent exercise on small EV microRNAs and then the pathways that they would target. And what we saw there was, compared to before training, 1 week of training actually leads to reductions in inflammatory pathways. So the well-known anti-inflammatory effects of exercise very well may be mediated in part by this change in microRNA that then can be communicated through paracrine and endocrine fashions through muscle-derived small EVs. So thoughts for the future. Clearly we see there's active crosstalk between muscle and endothelial cells. We've termed that Vasomyo Crosstalk. Whether we make muscle or endothelial cells dysfunction, it has reciprocal negative impacts on the recipient cells. That these small EVs, which are released by exercise and get into the circulation, can have both paracrine and endocrine functionality. That in addition to the microRNA that I've discussed here, we also know that these EVs contain proteins, lipids, and messenger RNA, and there's a large amount of research needed to be able to coordinate and integrate what all these species within these EVs, what do they mean and how do they work together. But at the same time, keeping in mind that while we have a lot to learn, it's very exciting because we could start to harness the powerful effect of these EVs, for example, following exercise training, as I showed you, to help reduce inflammation in target cells. And the interesting part about EVs is that EVs know how to bind and deliver content into cells. So they can be very cell site specific, we believe, as well as have this unique ability to be able to deliver content efficiently and effectively. Given that we're here to talk about the impact on social justice, it's critical that we think about how the injustices that occur across the United States and around the world, in particular for us, how they relate to cardiovascular disease. Because we know cardiovascular disease is the leading cause of death in the United States, and that the risks are higher based on race, ethnicity, and socioeconomic status. In fact, 25% higher cardiovascular disease death risk in our Black population, 50% higher hypertension risk in Black populations, 25% higher obesity risk in Black and Hispanic populations, 50% higher diabetes risk in Black and Hispanic populations, all of these compared to white populations. And we know there's 100% higher cardiovascular disease risk in poor versus median to wealthy income populations. And it calls for us for further work, more research on risk factors to help us identify what is important to understand, as well as the mechanistic differences within racial, ethnic, and low SES populations. And one of the real exciting pieces where the NAK can impact, due to our breadth of scientists, from exercise physiologists, biomechanists, neuroscientists, social scientists, behavioral scientists, historians, clinicians, we can, through multiple avenues, be advocating for evidence-based public health approaches to prevent cardiovascular disease in racial, ethnic, and low SES communities. And these approaches need to include lifestyle approaches such as increasing access to physical activity and increasing access to healthy foods. And when we can bring justice to these populations that we serve, then we will be able to fulfill our true missions. I want to thank the students who've worked in the lab over the years. I also would like to thank our funding agencies who, without their support, we would be unable to do much of our work. And I'd like to leave you with this. Healthy muscles plus healthy vessels equals healthy living, because that's what we want for everyone. And I know that's not just my sentiment, but of my colleagues here in the NAK. Thank you for your time. I welcome any questions you have, and feel free to reach out to me at any time if I can ever be of any assistance.